Okay, so next we are going to talk about the technologies or method to study 3D genome structures. So of course, ideally, if we can really see everything with our own eye, that would be ideal, right? And we have made some progress in that end. So as mentioned in the beginning, there's a chromosomal territories in the genome. So this paper from 2005, in this work, scientists began to label each chromosome with different color of dye. And when you combine everything together, you can begin to see these beautiful chromosomal territories inside the human cells. So you can see chromosome 1, 18, 20, and then you can see there's one copy of chromosome 18 here, and here's another copy of chromosome 18, right? So in the human genome, seems that these two alleles, they are not located together. Right? We can further zoom in, and then a very common technologies used to study chromatin interactions is called FISH, stands for Fluorescent in Situ Hybridization. So we can use different colors, even 2, 3, or 4 colors to do FISH. So the idea is like this. Suppose you have two regions in the genome, say in this case L2 and L1. You want to investigate whether there's chromatin looping events between L1 and L2, right? So all you need to do is you just use different color labels to label that strand of DNA. You just need to denature the double-stranded DNA and put the label there, right? And your control is usually, because you are investigating L2, usually you go to the other direction and you label the L3 with another color. So the idea is the genomic distance between L1 to L2 and L3 to L2 is relatively the same, right? And then you do a FISH, and then you observe them under the microscopy. So in this case, you can see actually in this case, the red and green, these two dots, right? They are located close to each other, meaning they are really close in the 3D space. Right? But this sort of light blue-ish color, although they have the same genomic distance, is located pretty far away from the red and green spots, meaning the chromatin action or the looping is only between the L1 to LT. And then there's no, and L1, L2, and L3, they're pretty far away from each other. Right? So one potential limitation for FISH is actually you really need to know which element you are investigating. Right? So in this case, because you need to know the location for L1, L2, so that you can design your probes. So the other thing you also need to know is FISH is still relatively low throughput. Each time you only investigate one pair of loci, and then the resolution is also not ideal, usually tens or even hundreds of kbs, right? Okay. So about this 2002 science paper, Dr. Joop Dekker invented this technology called the 3C, which stands for chromosomal conformation capture. So this has really created a new field in the study of 3D genome structures. Right? Actually, now we're looking back, the design is quite simple and straightforward, but I think this is really groundbreaking. Right? So inside your cell nucleus, suppose this is a strand of DNAs. So although they are located far away from each other, they are captured by this protein. So they are close in the 3D space. So what you need to do is you cut the DNA with restriction enzymes. They can be 6 base cutters or 4 base cutters. And then sometimes they have this sticky end, not evenly cut, right? So you cut your DNA, you will have product like this, right? And then in the next step, you just need to induce ligations so that this part is ligated to this part. And in the last step, you do the reverse cross-link, remove the proteins, and you have product like this. So ideally, this is from a different part of genome. And all you need to do, you just need to design primers and then do qPCR. Have these interaction frequencies. Right? So high C resolution, as you can tell, because you design primers to do a qPCR, is really base pair resolution, right? Give you high resolution results. But again, the potential limitations for this is, again, you need to know the region you are testing, right? But sometimes you do. In this paper, Nature 2012, is back then when I was a postdoc in Bing's group. In that paper, we were able to identify or predict there's an enhancer on the right side. They are enriched for histone modification, H3K27 methylation, right? We predict this enhancer regulate this gene on the far left, so across almost like 200 kb, right? And we suspect this enhancer regulate this gene only in cortex, in the mouse cortex. Therefore, my collaborator Yin actually she did a 3C experiment in both the cortex and the brain. So when you do the 3C experiment, you usually design a series of primers surrounding your targets. So on the right hand, this is your anchor point. On the left hand, this is the walking primer, right? You really have 5 or more primers. And this sharp peak right above the gene promoter, suggesting that there is a interactions between this promoter to this anchor point. Right? So this is a 3C experiment. So 3C is still widely used in different in current research. Okay? And then the really groundbreaking discovery is made from same group, Job Decker's group. He in 2009, right? I think he collaborated with Eric Lander's group. They were able to greatly expand the 3C experiment into called Hi-C, which is really high throughput. So in one experiment, they can give you the genome-wide chromatin interactions. And because it's taking advantage of the sequencing platform, right? So in the beginning, you can see this plot is very similar to the 3C experiment, right? You cross-link your DNA, you cut them with the restriction enzymes. The only difference here between 3C and Hi-C is actually here, when you fill the end, you add a biotin, right? And then the following step is pretty much the same. You ligate your DNA and reverse crossing and then share DNA, bring them into fragments. Only here, because when fixing your end, you add biotin. In this case, you can use antibody. You pull down the fragments containing the biotin, and then you can sequence libraries that only contain the biotin fragments, right? And then you are hoping that because when you do a sequencing, you use pair-end sequencing. You have one end coming from one part of genome, and the other end come from the other part of genome. And then this is, once you have the sequence results, potentially millions of reads or even billions of reads, you can begin to construct the data matrix, okay? Again, when you process in the ChIP-seq or ATAC-seq data, your data is one-dimensional. That means actually you can even have base pair resolution, is one dimension. But when we talk about the Hi-C, you are talking about two loci interaction with each other. So it's a n square, right? In that case, human genome is 3 billion base pair. You cannot do the base pair chromatin interactions. That would be 3 billion times 3 billion, right? So the result, when you process Hi-C data, you put data into different bins. Of course, the deeper sequencing depth you have, you can use a smaller window, right? And then as a result, so suppose this is your 1 KB window, this chromosome 1, 0 to 1K, 1K to 2K, blah, blah, blah. And then your y-axis is the same thing, right? You are investigating the chromatin interactions within chromosome 1, right? So each data value here, for example, 7 means there are 3 reads. One end mapped to the genome loci 0 to 1K, and the other region mapped to the region 3 to 4K, right? And then in this case, here, this spot, you have 10 reads, one end mapped to 1K to 2K, the other end mapped to 6K to 7K, right? So you can tell, right, the stronger value or bigger number you have, potentially there's strong chromatin interactions between this loci to the other loci, right? So we're good? Good. If there's no questions, I'll keep continuing, right? All right, now you can convert this data matrix into the beautiful heat map that you use to see, right? Just given different shade of red or purple, whatever your favorite color, right? So in this case, you can see in case the strong red dot here means the same thing. There's chromatin interaction between this loci to this loci, right? You may notice that this is we are investigating chromosome 1 to chromosome 1, so that's why the data is symmetric, right? So this is the first figure that draw in this Hi-C paper, Science 2009. So what they are showing here is the chromatin interaction for the whole chromosome 14, right? So immediately you can begin to see there's a clear patterns, right? A lot of the data analysis pipelines or algorithms is really trying to dissect Hi-C data matrix and looking for patterns, right? So this is one way to representing Hi-C matrix as a square, but sometimes you can also see the triangle-like shape. So what did it do, right? So essentially you can imagine just this square, you can rotate it to 45 degrees, have something like this. And because Hi-C data is symmetric, so now you can only see the half, right? So essentially this square and this triangle, they're essentially the same. Just people have different preferences. Some people are more familiar with this triangle-like structures. The only difference here now is each dot in the Hess mapping triangle indicating interactions between this loci and this loci, right? Has nothing to do with the loci right below it, right? So initially, I take this as for granted, and then later on, some people ask me is what's the dot stand for? Okay, in this triangle-like structure, dot here means the interaction between this region and this region, and you can begin to add your favorite genome browser shots here, right? So you can directly linking the genes or your his modification chip-seqs with chromatin actions on the genome. All right. So one feature you can observe is actually the closer you are to the diagonal, right, the values are higher, right? So in this case, the plot, the x-axis is the genomic distance between the two loci, and the y is the contact probability. So essentially that means the closer the two genomic loci to each other, they tend to have chromatin interactions, which is relatively intuitive, right? So imagine, so this is your DNA, right? The idea is that when the two parts are close to each other in genomic distance, it's easier for them to form a loop and contact each other. But at this long end, here to here, you need to form this much bigger loop, right? So the chance for this is really smaller. So this is what we call a distance decay in the Hi-C data sets. Okay.